My name is Jain, and I'm here on behalf of Sapient. And I just want to thank the AAPS and the audience for the opportunity to present some of our ongoing work today from Sapient, in which we leverage next-generation mass spectrometry systems to accelerate biomarker discovery and drive drug development. Tackling a problem that's well known to many of you. And that is the failure rate that's inherent in drug development is extremely high, estimated to be over 96% for all drugs that are under development. The reason for this failure has to do with a fundamental mismatch between the way we diagnose disease, which is based upon an end pathology, and the way we develop drugs, which is targeting an underlying mechanism of action. Now, this mismatch between the way we diagnose and the way we develop drugs is what gives rise to this high failure rate. Now, of course, this mismatch has associated with it a number of challenges and risks, including risks to patient safety, risks to time and the amount of duration it takes to develop drugs, and of course, the great expense associated with drug development today. Many of you are aware the diagnosis of disease hasn't fundamentally changed in thousands of years, going back to the days of the ancient Greeks. Shown here is the underlying urine wheel on the right, in which urine was assayed for particular odor or particular color or even particular taste, and these chemical changes were used to diagnose disease. Fundamentally, we do the same thing today. This mismatch between diagnosis and drug development? Well, the answer to us is through the use of circulating biomarkers. Biomarkers provide specificity at scale and allow us to align the right patient with the right disease with the right therapy. A tremendous amount of data to support this underlying idea. If you look at the tens of thousands of human clinical trials that have been done over the last 2 decades, those clinical trials in which biomarkers were used had an approval rate by the FDA that was 2 to 10-fold greater. In addition, not only is the approval rate greater, but the time to approval is actually over 20% faster. Now, this is across many different areas of drug development, whether it be oncology, cardiovascular disease, vaccine development, infectious disease, et cetera, et cetera. And if we look at all clinical trials, over 20,000 of them, across the summation of these trials, there's over a 2-fold increase in actual FDA approval when a biomarker is used in the drug development process. Well, why is this? Well, biomarkers allow us to identify and confirm the right mechanism with the right target, the right safety profile, the right pharmacometrics, and ultimately the right patient, beginning to align the underlying patient with his or her disease process and his or her specific therapy. To date, the vast majority of biomarkers for human disease have been decoded through human genetics and next-generation sequencing. The challenge is that genetics alone, however, is not sufficient to decode human disease. If we look across a number of diseases, whether they be cancer, heart disease, neurologic diseases, lung disease, autoimmune disease, et cetera, what we find is that genetics accounts for a minority fraction of risk, somewhere in the order of about 15 to 20% of population with attributable risk for any of these singular diseases. What this means is that 85% of risk factors and subsequent biomarkers for any given human disease still remain unexplored. Population attributable risk is not encoded in your underlying genetic sequence, but rather is encoded in small molecule biomarkers that exist in human circulation. These biomarkers read out internal organ physiology and structure, environmental exposures such as diet and drugs and toxicants, as well as internal milieu, including the microbiome. Now, traditionally, these small molecule biomarkers are captured and assayed using mass spectrometry. The challenge today is that current mass spectrometry approaches are too slow to be leveraged on a population level scale for biomarker-based discovery. spectrometry profiling at scale. We have developed, optimized, and standardized next-generation proprietary rapid liquid chromatography mass spectrometry, or RLC-MS-based systems that allow us for capturing tens of thousands of molecules in a single human biosample in under 1 minute. We have built one of the world's largest capacity bioanalytical facilities that allow us to handle and analyze, on average, more than 20,000 human biosamples per day. These are leveraged and integrated within deep biocomputational expertise that allow us to use everything from regression statistics to more advanced machine learning tools to identify the key circulating biomarkers of human health and disease. These are coupled together with a very unique and proprietary human longitudinal disease database that is the world's largest human chemical database to date, encompassing over 300,000 humans, followed for over 30 years, 20 million patient years of adjudicated outcomes data. Together with our biocomputational learning and human disease biology database, allow us to rapidly discover actionable insights. Starting from a population of humans in human biosamples in which tens of thousands of markers are assayed, to identify singular markers and then determine the specificity of these markers in an underlying database across hundreds of thousands of individuals. Sapient learned by using these technologies. Well, first, across large human populations, there is remarkable diversity in circulating biomarkers. Across 50,000 individuals, we find approximately 200,000 different molecules in circulation. Among these molecules, somewhere in the order of about 12,000 molecules are common, meaning they're present in more than 50% of the population. Another 3,000 to 4,000 molecules are universal, meaning they're present in virtually everyone, irrespective of one's lifestyle, location in the world, age, etc. These common or universal markers include those things such as glucose, common amino acids, common lipids, and the other molecules that we all need to live and walk and talk. Now, in any one singular individual, as shown in the top left here, there's approximately 10,000 to 12,000 different molecules. People at an advanced age tend to have slightly more molecules than people at a younger age. Again, in any singular individual, there's common molecules that are present across the population, as well as molecules that are more rare. Now, among those molecules that are present, their magnitude and concentration vary greatly across populations, as shown here in the bottom left. Certain molecules, once again, such as glucose, are defended against, and their levels vary little across populations. However, other molecules, yet undiscovered, vary tremendously by orders of magnitude from one individual to another. If we think about the dynamic nature of these circulating biomarkers, as shown in the bottom right here, we see that over a 24-hour period, if we sample blood every hour, that many of these molecules in blood change dramatically over the course of a day in relation to exercise and rest, fasting and feeding, sleep-wake cycles, and many, many yet undiscovered factors. However, if we look at the distribution shown here in blue, in which blood samples were taken at a fasted state in individuals and then repeated at a fasted state approximately 2 years later, we find that the vast majority of molecules change very little. In fact, more than 50% of the molecules change less than 20% over that 2-year period. This has to do with continuity of exposures, both in internal organ physiology, our microbiome, our diet, and our lifestyle. This is extremely important, and the stability of these underlying biomarkers, particularly in a fasted state, is what allows us to use these for predictive analytics and to understand human health and disease. Influences the absence and presence or the levels of these circulating biomarkers? Well, as perhaps would be expected from the great chemical diversity, the answer is rather complex. These circulating molecules are influenced by both genetic factors, environmental factors, as well as shared environmental factors. When we look across a variety of underlying exposures, we find that the human microbiome contributes importantly to these circulating biomarkers. If we think about common demographics, lifestyles, or even laboratory values, we find that all of these contribute in some way, shape, or form to circulating biomarkers, but overall, they influence the underlying levels of these markers to a minimal degree. The underlying diversity among all of these circulating biomarkers is quite great across whole populations. We find common molecules, once again, including amino acids, bile acids, glycerol phospholipids, fatty acid molecules, as well as many, many other well-known chemistries. However, when we look across the population, across these tens to now hundreds of thousands of circulating factors, we find that the vast majority of them actually represent novel chemistry that has never been mapped anywhere in the underlying world. Sapient and its underlying approaches have been used by a variety of organizations, including the biopharmaceutical industry, foundations, the US government organizations, including the National Institutes of Health, as well as foreign governments for a variety of different applications to answer many different questions in human health and disease. Example by which Sapient and its technologies have been used to identify biomarkers of disease progression. As many of you know, within a given disease, there's oftentimes individuals who decline at a rapid rate, whereas other individuals with the same underlying disease have a much more slow and indolent progression. Understanding which batch individuals fall into have important implications for both disease understanding, for drug development, and for clinical trial optimization. Here's an example in diabetic kidney disease in which Sapient's technologies were used across 100,000 human biosamples over time to identify a singular circulating biomarker which denotes those individuals who are going to have rapid progressive diabetic kidney disease with an odds ratio almost tenfold greater than individuals who had low levels of this underlying circulating marker. This singular biomarker allowed Sapient to work with its client to substratify patients according to rapid progressors. It's being developed as a companion diagnostic to drive clinical trial optimization. And most importantly, this singular marker reveals unique pathways essential to rapid disease progression. By which Sapient and its underlying technologies may be used to identify biomarkers of drug efficacy, essentially stratifying individuals into drug responders and drug non-responders. Now, of course, there exists across any individual disease state a great degree of heterogeneity and variable response to drug therapy. In one such example, Sapient was able to take a population of individuals that were exposed to a drug and find a very unique pharmacodynamic biomarker that elevated in some individuals exposed to the drug, but not other individuals who received the same exact dose, irrespective of pharmacometric measures. Study, those individuals that had an elevation in this pharmacodynamic biomarker responded wonderfully to the drug, whereas those individuals that did not have an elevation in this pharmacodynamic biomarker had poor response to the drug. Sapient was able to further take this underlying marker and study it across thousands of individuals from our underlying longitudinal human database. And in doing so, we found that individuals with a high level of this specific marker are more likely to die from this specific disease relative to individuals with a low level of this marker. Longitudinal database, we were able to even do genome-wide association studies for this singular marker, and in doing so, identify key biosynthetic enzymes and potential therapeutic targets. This specific biomarker represents a unique pharmacodynamic marker that allows us to stratify patients into responders and non-responders. It is currently being leveraged as a companion diagnostic to guide clinical trials. And the underlying biosynthetic enzymes that were identified to give rise to the specific markers are now being developed as new therapeutic targets. Approaches allow us for the first time to achieve true specificity at population level scale. These approaches may be used for a variety of different questions and problems, including target identification, detection of early disease states, understanding of disease progression, assessment of target engagement, patient stratification and companion diagnostics, safety profiling, pharmacometrics and quantitative systems pharmacology, and even clinical trial optimization. Thank you so much for your time and attention today, and please don't hesitate to contact us if we can help you discover more. Shown below here are a phone number, an email, and a Calendly link that can be used to schedule a meeting or visit at our booth here today at the AAPS. Thank you so much.